Doc here with a review of Chapter R, The Human Ear, where we focus on physics and biology. So let's go to the text, the PDF file. So we go to Downloads, and in Downloads here, we're going to go to the Foundational Physics, R, and here we go. And in our course, we look at the human ear with poetic license in terms of the biology so that we can emphasize the physics. So we do not make the ear look precisely, very precisely like a biology book would do. In other words, more like a dissection. We here do a sketch to show the general features, and we have the shapes looking roughly approximately correct, but we do not emphasize biological structure in detail as much as we do the physics. So let's look at this ear, and before we do that, I'll show you a more accurate biological structure here. And in this little applet, very nice here from Ireland here, we're looking at the eardrum vibrating as the sound comes in the ear canal. Here you have air, and here you have the bones, three bones in the middle ear that are moving, and then you have fluid, say, in the inner ear. And this is nice, and this shows you more precise, biological structure, which we're going to simplify and isolate. Notice here we have different frequencies that we can do here. And you have your outer ear, your middle, and your inner. So let's look at that in a rough sketch like we have here. This is your outer ear, everything from the outermost part to the eardrum. The eardrum is your boundary between the outer and the middle ear. Then the three bones, the hammer, the anvil, and the stirrup. And then the boundary between the middle ear and the inner ear, the oval window. Notice this is your eustachian tube, and then in the inner ear you have the cochlea. You also have balance sense here that's near the ear, but we're not going to talk about that. That's where you get dizzy if you spin around. So here we look at these three regions: outer, air conduction, middle ear has bones that move, solids, and here you have fluid in the inner ear. So we come down here to the cochlea to unwind it. So we take this here in the inner ear, the cochlea, and unwind it. And when we do that, we get here the oval window that you see up here, oval window, and the round window, and we unwind this. We're going to then orient it left to right. And we have the scala vestibuli, a chamber up here, and a scala tympani, and the main support here is the basilar membrane. We'll look at this in more detail. And think of this as having different stiffness values so that it's more wobbly down here but stiffer up there. And if you have a stiffer thing, boing, that will vibrate at a higher frequency. So when the frequencies come in, the higher frequencies get picked up here. And you can think of the pathway as going like this. This is a pressure place to give. If there's excess pressure coming through, so the sound wave do something like this. If you have a low pitch, the low pitch gets picked up here where it wobbles, more wobbles here, it's going to vibrate at a higher rate. So your frequencies along here with your 20 hertz down here and your near 20,000 hertz up there. So let's look at the auditory nerve that goes off to the brain. We simply slice this, and the auditory nerves here fan out. Now you have to coil that up, roll that up in the final version to put it up in there, but we need to unwind it to study it in more detail. So here we have the cross section. Cross section just means slice this thing and then look at down the tube. So we do that. We see the scala vestibuli at the top. We have the scala tympani down here, so sound comes in here, crosses over depending on the frequency, comes out here, and the auditory nerve takes the signal to the brain. So what about this middle region here. What about this line here that goes across here? Let's blow that up. That's where the action takes place. We blow that up. We have a protective membrane, the Reissner membrane, under here the scala vestibula, the top chamber. Then we have the tinctorial membrane, which will tickle the hair cells, some 20,000 of these that reside on the organ of Corti, which then resides on the basilar membrane, which has the different stiffness values. So here, if it's being stiffer here than it is to the right, that you get action here, vibration here, the sound then crossing over from the top down to the bottom. So oval window, cross over here, little tickling effect off to the brain with the signal, and you got it. The detection of sound. What about this value or arrangement of the frequencies? We said that over at the far right you have the low frequencies and here you have the high. So let's start here with 20. By the way, this length of the cochlea is 3.5 centimeters. 1 centimeter is the width of your little finger, roughly. So that's pretty small. And that distance of 3.5 centimeters covers all of the frequencies we can hear because the numbers are arranged in a non-linear fashion. That means every time you take a step, you don't add. This is linear. I take a step, I get say 20 hertz or $20, I have $40. I add 20, then I add 20, 60, 80, 100. Look, you'll never get all the frequencies detected in the ear that's that small with that kind of a scale. So we use the doubling scale and non-linear scale where you double. And remember, when you double, that's 1 octave. So do, re, mi, fa, sol, la, ti, do. See, that's the other do there. You double, you get 40, then 80, 160, 320, 640, 1280, and then here 2400 plus 160, 2560, and here 2500 double is 5000, and 60 doubles 120, 5120, and then here this one's easy to double, 10240, and then here 20, and then 4, 8, and 0. So the range of human hearing given from 20 to 20,000. And let's look at these here. Count the octaves. Do, re, mi, fa, sol, la, ti, do, 1 octave. Do, re, mi, fa, sol, la, ti, do, 2 octaves. 3, 4, 5, 6, 7, 8, 9, 10. You can hear about 10 octaves. 9, easily 9. Folks lose it up here around 18,000 or so. If you get older, you tend to lose these high frequencies. So that's 9 octaves, and compared to light, if you look at red light, the frequency of red light, if you double that, you're past the blue, violet. It's like you only see 1 octave of color, but you see multiple octaves of sound. And with color, when you put 2 colors together, they blend in, like 2 paints get blended in. You see 1 color, but when you hit 2 notes, you can tell there's 2 notes there. A violin's playing 1 note, and say clarinet's playing another, or the piano's playing a chord. You can tell there's more than 1 note being played. So the ear does that spectral analysis and has many, many octaves there. So here, we're looking at a way of looking, say, of abbreviations here, rounding things off. We're saying here, look, 30, double, 60, 125 is a nice number because you double, that's 250. So you kind of nudge the numbers a little bit. 500, and then you double that, you get 1,000, 1K, 2K, 2,000. Double that, 4K, 8K, 10, 16K. 4K is 8K, 4, 8, and a 16. So 1, 2, 4, 8, 16. So this is a way to write the numbers and to see what's going on, and that's actually more realistic, higher pitch that folks can hear. So here we're going 1, 2, 3, 4, 5, 6, 7, 8, 9 octaves. So since this whole distance is 3.5 centimeters, that means each octave is a tenth of that. So a tenth of that would be 3.5 millimeters. 3.5 millimeters for each of those octaves. Well, Look at our equalizer. Did we use a similar principle where we had, say, 60 and double, get 120 or make it 125, nudge it so that here you get 250, 500, 1000, 1K, 2K. See, that's the same principle that's going on here in the ear, and it's no coincidence why we would do it here for the equalizer.